everyone. Thank you for coming and for joining by your presence here tonight in KAUST's quest to build a 21st century Beit El Hikmah, our House of Wisdom, right here on our Red Sea Resort campus. An institution that's worthy of comparison with the original 9th century Beit El Hikmah, which was noted for collecting, translating into Arabic, furthering and disseminating all of the scientific and technological knowledge in the world at that time. When we opened Winter Enrichment 10 days ago and committed ourselves to the pursuit of wisdom through the integration of knowledge, you most likely assumed that the knowledge you would be receiving would be coming from leaders in science, engineering, commerce, and arts. Did you think that you would be learning from clams and snails? Actually, products of natural evolution represent an incredible source of insight for engineering design. For them to be here in their current forms, they are optimized for something. We have also talked in WEP 2015 about detecting and pushing our limits. What are the physical limits to climbing, swimming, sensing? And practically speaking, can we design better engineered systems for instance, the oil industry, the transportation industry, the building industry, or others, by studying the genius of nature in pushing these limits. In the next hour, Anette Hosoi will dip into her years of deriving personal inspiration from relatively simple organisms and bring up some interesting examples. I'm sure we will enjoy them, as have countless other audiences around the world. Pekko is professor of mechanical engineering at MIT. She received her BS in physics from Princeton, her PhD in physics from the University of Chicago in 1997, and then postdoced as an NSF fellow in the MIT Department of Mathematics and at the Courant Institute of Mathematical Sciences at NYU. She is a leader in the study of hydrodynamics of thin films and in nonlinear physical interaction of viscous fluids and deformable interfaces. Her work spans multiple disciplines, as you can guess from her degrees, including physics, biology, and applied math, and it's being used in a number of industrial contexts, including collaborations with Schlumberger, Bluefin Robotics, and Boston Dynamics, to guide the engineering design of robotic crawlers and other mechanisms. She is also, and this can, I think, be the subject of a future KAUST lecture, the founder and director of STEAM, the Sports Technology and Education at MIT, which is dedicated to building a community of faculty, students, industry partners, and athletes who are passionate about tackling challenges that lie in the intersection of engineering and sports. As you're about to enjoy, Professor Hosoi is an innovative teacher. In addition to her many teaching awards, she is the recipient of the 3M Innovation Award and has held the Doherty Chair of Ocean Utilization at MIT. Also a Radcliffe Institute Fellow, she was elected to fellowship in the American Physical Society in 2012 and selected for the Siam Block Lectureship in 2013. Pekko, welcome.
Thank you. Thank you for that lovely introduction, and thank you for inviting me to participate in this wonderful program. I've had a really inspiring 3 days, and I know I'm going to bring a lot of new ideas back with me to MIT. So today I'm going to talk to you about mathematics and bio-inspired design, and I thought I would start by just giving you a few of the most famous examples in the field. So starting with this guy right here. So many of you are probably familiar with the story of George de Mestral, who went walking with his dog in the Alps, and when he came back, his dog was covered in these guys. And after spending many, many hours picking the burrs out of his dog's fur, he thought, gosh, this would make a great fastener, and this, of course, led to the invention of Velcro. But not all of the bio-inspired design stories are so successful. So in addition, people have spent centuries looking at birds, thinking, gosh, it would be great if we could build some kind of a flapping machine that would enable us to fly. And what this has led to is hundreds of hours of YouTube videos that look like this. So, okay, so the question you have to ask yourself if you're going to work in the field of bio-inspired design, oh wait, I have to wait because this is my favorite guy in this video. Okay, there's a point right here where you can tell he's really sure this is going to work right here. Yeah, no, okay, okay. So, all right, so I have to admire that because, I mean, he really went for it, right? But the question you have to ask yourself is, okay, if you're going to work in the field of bioinspired design, how do you make sure that you are designing Velcro and you are not just making hundreds of hours of YouTube video that looks like this? And there are many, many answers to this question, and I'm going to start by just giving you some guidelines that we use in our lab. So in our lab, to pick the sort of problems that we think are going to be fruitful, we have a couple of guidelines that we follow. The first is that I'm only interested in choosing simple organisms to emulate. Okay, and so in particular, I want organisms with a very primitive central nervous system, preferably no brain, because brains are very, very hard to build. And I'd rather build something where the solutions lie in mechanics rather than in a central nervous system. Second, I'm interested in organisms that in some way are orders of magnitude better than what we can currently do in engineered systems, right? So they have to be better in efficiency, in robustness, in versatility. In some way, they have to outperform existing devices. And the third one, and this is probably the most important piece, is that the goal here is not to just copy nature, because if you copy nature, again, you're going to end up with that YouTube video. What you really want is you want to understand the underlying physics, mechanics, or dynamics that enable that biological solution, and then use that to guide engineering design. So those are going to be some of the themes that you're going to see in this talk. Now, there's one big assumption that underlies everything that I've said so far, and that is that for some reason nature is worth emulating. And the reason you might imagine that natural systems are worth emulating is that because they have had millions of years to evolve, many of them have come upon, or I'd say have converged on optimal ways to perform certain tasks. So we as designers and engineers and inventors can learn a lot by looking at those natural systems. Okay, so that's one, is to guide engineering design. There's another reason optimization is interesting in biology, which is a little bit more subtle, and is a purely scientific reason. And I think it's nicely summarized in this essay by William Sutherland that appeared in Nature in 2005. And I'm just going to read the opening paragraph of this essay that he wrote. Okay, so in this essay he says, right, if I start here, there are increasing calls for biology to be predictive. Optimization is the only approach biology has for making predictions from first principles. The wider adoption of these ideas right across biology should reap ample rewards. So this is, in fact, a somewhat controversial statement because of the word only right here. And I'm not going to debate that today, so I invite all of you after the talk to think about whether this is only should really be here, or if you can think of other ways that biology can be predictive. But what I think is certainly true is that optimization gives you a mechanism to rationalize the structures and strategies that you see in natural systems. Right? So there's a purely scientific reason to actually think about these kinds of optimization problems. Okay, and then of course you can design, improve engineering design. So optimization is going to be another theme that you're going to see in the talk. So I thought I would start by just putting up the definition of optimization. And this, I literally Googled optimization, and this is the definition from Merriam-Webster. Okay, so according to Webster, optimization is an act, process, or methodology of making something as a design system or decision as fully perfect, functional, or effective as possible. Specifically, and here I've highlighted this, the mathematical procedures as finding the maximum of a function involved in this. Okay, so this is optimization. So for example, suppose I have a function, so there's a function, and suppose I derive this function by making a model of something that I'm interested in. So maybe let's suppose I'm thinking about swimming fish. So suppose this is the function that tells you the swimming speed as a function of the undulation frequency of the fish's tail. And I said, please find for me the optimal solution. And you say, oh, that's very easy. The optimum is right there. I just have to find the maximum. And so this is the frequency that I'd want to use if I'm a fish. So that's all fine. The problem is that biology rarely gives you functions that look like this. Unfortunately, biological systems tend to give you functions that look like this. They're a huge mess, right? And so if you look at something like this, it is much more challenging to find optimal solutions. And this, I've drawn this in 2D. In bio, it's actually worse because they're in 5-dimensional space or 6-dimensional space or 7-dimensional space. Okay, so the question of finding optimal solutions is A, incredibly useful, and B, incredibly challenging. And because of those two things, most of the great minds in applied mathematics have thought about this problem. So let me give you just a brief history of optimization. And that the history starts actually with Fermat. So this is Fermat of Fermat's last theorem. And so he, in 1638, wrote this Methodus ad discurrendum maxima et minima. And the thing that I find most remarkable about this is that Fermat wrote this paper 50 years before calculus was invented. Right? So imagine that, those of you who have taken calculus, imagine I give you a function and I say, please find the maximum, and you say, oh yeah, that's easy, I'll just take a derivative, I'll set it equal to 0. And then I say, no, wait, calculus hasn't been invented. Please find it without using a derivative. Oh, and computers haven't been invented either, so you can't use that. Okay, much harder problem. All right, so the next person who thought about this is Newton. So, of course, Newton must have thought about this. And Newton, interestingly enough, even though he was one of the co-inventors of calculus, also did this without calculus. So he also noticed this is when calculus was invented. His paper also appeared before the calculus papers appeared. Okay, and then if we move forward, we get to Lagrange. So Lagrange did something called the Euler-Lagrange methods, which I'll talk about a little later. Gauss, of course, Gauss. So he did an iterative method. And von Neumann, again, looked at iterative methods in solving these problems. Okay, and you can see there's one more slot here that I'm going to put someone in. But before I put that person up, I want you to take a look at this list. Okay, this list, this is the pantheon of applied mathematics. These are the greatest minds to have thought about applied mathematics in the past X centuries, whatever it is. So I think it is very, very dangerous to put any modern day person on this list, right? It's very dangerous. Okay, so, but I'm going to put someone there, and I think the person that deserves to be there in the context of optimization is George Danzig. Okay, and George Danzig, so I didn't know anything about George Danzig before I started writing this talk, but the more I learned about George Danzig, the more I liked this guy, to the point that I'm now going to put up a slide on George Danzig to tell you guys about what he's done. So George Danzig, I'm going to start with a story which is possibly apocryphal, but might be true, and it's such a good story I'm going to tell it anyway. So George Danzig was a graduate student at Berkeley, and he was a good student. Not surprisingly. He was also not a terribly punctual student. So there was one day when he was going to his statistics class, and he came in 10 minutes late, as he normally does to his statistics class, and he did what everybody does when you come in 10 minutes late to a lecture. You sneak in the back, and you sit down, and you get out your notebook, and you start frantically copying down the notes that are on the board, and he also copied down the 2 homework problems that were on the board. You know, you catch up, and eventually you get back in sync with the lecture. So he took those notes, he went home, and he worked on the homework, and then he turned in the homework late, apparently, a week late or something, but he turned it in. Okay, so fine. So he turned in the homework late. And then about a week later, there was a knock on his door at home. And he opens the door, and it's the professor of that class. And the professor of the class is there knocking on his door because the 2 problems he solved were, in fact, not homework problems. They were examples of the 2 greatest unsolved problems in statistics. And so this turned into George Danzig's thesis, which was a great thesis. And when he was interviewed about this later in life, he said, yeah, I did think those were a little harder than the usual homework problems. Okay, so that's why everybody loves George Danzig. So Danzig, so then after he graduated from Berkeley, he joined, he worked for the government. He served as a mathematical advisor to the Department of Defense. Okay, and he did this in the 1940s, so this was right around the time that World War II was happening. And World War II, of course, there was a tremendous logistics problem about moving people and equipment and supplies around. So that's the problem that George Danzig worked on. And he invented something that was called linear programming. So in this case, so programming does not mean what we normally think it means, because again, this is 1940s, so computers have not been invented yet. Okay, so programming in this case means logistics. Okay, and so linear programming was, as Danzig himself described it, provides the ability to state general objectives and to find optimal policy decisions for a broad class of practical decision problems of great complexity. Okay, so that's what his goal was. And the first problem he tested this out on was the following. What is the cost of the least expensive adequate diet? Okay, so this is something the military is obviously going to be very interested in, because you want to know how much it costs to feed your army. Okay, so they wanted to know what is the dollar amount to feed 1 person for 1 year. Okay, so 2 things I want to point out. So throughout this talk, when I pull up optimization questions that we're going to think about, I'm going to do it in this format. So anytime you see the red box, that's the optimization question we're thinking about. Okay, and second, I want you to think about what the answer is. All right, so I'm looking for a numerical answer here. I'm going to tell you a little bit about it, but in a minute I'm going to ask if you all have your answer in your mind before I put the actual answer up. Okay, so what Danzig did, he wrote down the system. So this was a system of 9 equations with 77 unknowns. And again, there were no computers, so this took 1,000 man-hours to calculate using calculators. Okay, but they came up with the answer. So does everybody have your answer in your head? I'm going to put this up in US dollars. Okay, everybody, this is the total number of dollars it costs to feed one person for 1 year. Okay, imagine how much it costs to feed yourself for 1 year. Okay, the answer is $39.69. Pretty cheap. Anybody get it right? Yeah, okay. Yeah. So this, and in fairness, this is in 1945 dollars. So in today's dollars, it's about $500. So that is how much it costs to feed one person an adequate diet for 1 year. Okay, so this is my introduction to George Danzig and to optimization, and I'm now going to move on to sort of 3 vignettes, 3 short stories about problems that we've been working on in our lab, which lie at the intersection of bioinspired design and optimization. So the 3 I'm going to talk about are snails, clams, and swimming microorganisms. And I'm going to start with snails. Okay, so snails. So snails are actually very bizarre animals. So if you take a snail and you put it on the ground, it will crawl along the ground, and when it hits the wall, it'll crawl straight up the wall, and then when it hits the ceiling, it'll just keep crawling, and it'll crawl across the ceiling, right? So imagine you could build a robot with that kind of versatility, right? We don't have any kind of crawlers or climbers that can do that. So, so we got interested in using snails to inspire locomotion in robotics, and the way they're able to do this is that when a snail crawls, it secretes this thin film of mucus that sits between the snail's foot and the substrate, okay? And so the only way that the snail moves, the snail's foot never actually contacts the substrate. The only way it moves is it drives stresses in this thin film, which drive flows, which then push the animal forward. Okay? So when you drive these stresses, you can actually see this if you look at a snail crawling up a plate of glass or if you look at a snail in an aquarium. So it looks like this. This is actually a slug, not a snail, but they do the same thing. And so you can see there are these waves that move up along the bottom of the foot. So those are the waves that are driving stresses in that thin film to push the animal forward. Okay? And biologists have studied this for over a century now. So it was the early like 1909 or something when these papers started to appear. And so they've classified all of these different kinds of waves. So this, for example, they call a monotaxic direct wave because there's only one that spans the foot and it goes in the direction of motion. This they would call a bitaxic retrograde wave because there's 2 waves and they go backwards. Okay? And if you sneak up on a snail and you startle it, it will gallop. So this is a galloping snail. Okay, so I'm going to be most interested today in this guy over here, the direct monotaxic waves. Okay, so the work I'm going to tell you about was work done by my student Brian Chan. And Brian Chan was the first student that I supervised at MIT, and he came to me and suggested this problem. So he had been working on a problem involving water striders. So water striders are those little insects that sit on the surface of the water and run around. And so for part of this project, he had to collect water striders. And the way you collect water striders is you take a big bucket and you go to a pond and you scoop up a bunch of water and you get water striders, but you also get a bunch of snails. So he'd been staring at these buckets where he saw a lot of water striders, but he'd also seen a lot of snails. And so he started to think, oh, maybe there's something interesting with snails. So he came to me and he said, you know, I'm interested in studying how snails move. And I thought, well, okay, that's strange, but all right, fine. And I said, okay, I'll tell you what, why don't you go and just take 2 weeks to look through the literature and tell me what you find. Tell me what's been done, tell me what's still interesting, tell me what there is to research. He said, okay, fine. So Brian left, and he came back 2 weeks later, and I said, okay, so what did you learn? And he said, oh, I solved it. Like, what do you mean you solved? I don't even know what the question was. What do you mean you solved it? And he said, oh, I built a robot. And I thought, what the heck? But now, so you have to remember, Brian was the first student that I'd supervised, and at the time, I had not realized that if you give an MIT student a problem with insufficient constraints, what they will do is build a robot. So he built a robot. I have now learned that you have to put more constraints on these problems. Okay, so here, I built a robot. Here's Brian's robot. And it turns out actually that in this case, building the robot was exactly the right thing to do because we learned exactly what we needed from Brian's robot. So it's a robot that has, so it has a spiral that runs through that's confined between two plates, and there are aluminum plates that are free to slide up and down on the spiral, so when the spiral turns, it drives a wave on the bottom of the foot, just like you see in a real snail. And if we put that on a viscous fluid, it crawls. And it turns out it is using the stresses in the fluid to crawl, because if you take the fluid away, it doesn't do it. First of all, it goes about an order of magnitude slower, and it also goes backwards. Right? So it really is driving stresses on this viscous fluid to push it forward. So the reason this works is the following. So here's my snail. Here I'm going to blow up the bottom of the foot. So here's the snail's foot. So you see, here's the wave that we made on the snail's foot. There's a fluid in here. So imagine I drive a wave that's moving in this direction, right? So this bump is moving this way, and this bump is moving this way. Okay. So that means if this bump moves this way, this fluid here is getting squeezed, so you're going to get a high pressure over here. This fluid over here is being opened up, so you're going to get a low pressure over here. Okay, so that means that this high pressure is going to act normal to this interface. So this high pressure is going to push this way. This low pressure is going to pull that way. And so you get forces that look like this, which you can see now. If you add these two guys together, you're going to get a net force in that direction. So it's going to push the snail that way. Okay, so that's how this works. And then the next thing that we might want to do is you say, okay, well, imagine the following. Imagine I could prescribe the shape of that foot. So I can make any shape that I want. Let's call this some function, h of x. And now I ask you the following question. I say, okay, if I give you the shape of the foot, can you tell me how fast the snail moves? And the answer is yes. And I'm not going to go through all the mathematical calculation, but if you're interested in it, so I have references throughout this talk, which you can look up, and I'll also give my website at the end. So all the references are linked over there. But the calculation is actually very straightforward. Any sort of first-year fluid dynamics student could work through this. And you get a very simple answer. So this is the velocity of the snail. So now we're finally prepared to ask the optimization question. Okay, so the optimization question is the following. What are the optimal shapes of the foot that maximize the snail velocity? Okay, so for example, I could make my foot shaped like this, or I could have it shaped like this, or shaped like this. Okay, and again, remember, when I have this box, I'm going to ask you what the answer is. So start to think in your mind what you think the answer is. You don't have to do any of these calculations to get the answer. The physics I told you on the previous slide is all you need to know. I'll be fair about this. I'll give you all the information you need. Okay, so while you think about this, let me just say that this is now a much more complicated optimization question, because I'm not looking for a single number. So remember, last time I was just looking for $39.69. I wanted one number. Right now I'm not looking for a number. I'm actually looking for a function. Right? So optimizing for a function is going to be considerably more complicated, but it can be done. And this, you remember I mentioned before, there's my pantheon of optimizers. This is Lagrange was the first to think about optimizing functions. So there's beautiful theory behind how you actually do these functional optimization problems. Okay, so this problem, so all of this stuff up here I just put up there to prove to you that we did some math. You don't have to read through that. But think for a minute about what you think the shape is, because I'm about to show you. So the optimal shape is actually this one right here. It's this guy. Okay? And so we didn't know, before we did the calculation, we didn't know this. But once you see the calculation, it's actually quite obvious. And let me tell you why. So what you want is you want something very flat with something sort of skinny and pointy, separating the flat bits. And here's why. So if I look at this again, remember I'm making a high pressure over here and a low pressure over here. And then that pressure acts on this interface. So what I want is I want this interface to be as vertical as possible, so that pressure is pushing the snail in the direction that I want it to go, right? So you want this interface aligned so that all of that nice pressure pushes you in that direction. So I want this to be very, 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 very vertical. Second, I want this guy to come very close to touching down, because I want a large pressure difference between here and here, right? So I want this guy to build up a lot of high pressure, so this should come almost all the way down. And the third bit that you need is you say, okay, well, in this place where the foot comes very close to the ground, there's a lot of drag, so you waste a lot of energy. So anywhere that you possibly can, you want to pull the foot as far away from the substrate as possible, so you're not burning a lot of extra energy. So that's why you have something that's pointy that goes up, and then pointy and then goes up again. Okay? So that was great. So we did this calculation. We found optimal solutions. And then from there, you can actually make this calculation much more interesting, much more complicated. You can think about different kinds of fluids, like yield stress fluids, things that look like mud, things that look like mayonnaise. And it turns out that if you're thinking about yield stress fluids, Schlumberger becomes very interested. because they use these kinds of fluids in their drilling mud. So the drilling mud is a yield stress fluid. And so we then built another prototype which crawls on yield stress fluids. And we subsequently made an axisymmetric version, and we passed this on to them, and their engineers in Japan are now actually using this design to build crawlers for downhole environments. So from snails to oil robots, that's the path that you take. All right, so that's my snail story. So next I'm going to tell you about clams. Okay, so here's clams. So clams, again, this started with a collaboration. Well, actually, this is a little bit different. This started with a collaboration with a company, and the company that we were working with is Bluefin Robotics. So Bluefin Robotics makes small underwater vehicles, and a small underwater vehicle is about 15 to 20 feet long. Okay, so this is one of their underwater vehicles. And what they use these for is they send them down, anywhere you want to monitor things, and they measure things like pressure and temperature and salinity, anything you want to measure, and they do it for long periods of time. So you keep them down there for something like 2 weeks. And the problem is that if you take one of these guys and you set it down there for 2 weeks, they tend to drift because there's currents, right? Okay, so they drift around. So what they would like is they would like the thing to stay in one place so you can get better measurements. So one thing you can do is something called station keeping, where the thing is smart enough to swim back to where it's supposed to sit. The problem is that's actually a fairly complicated controls problem, and it takes a lot of energy because you're constantly moving around and adjusting your position. So what they wanted was an anchor. So we said, okay, yeah, we're mechanical engineers. We should be able to make an anchor. How hard can that be? And then they said, okay, well, but here's the problem. So typically when you make an anchor, it's a giant weight that you just throw out of the boat when you're ready to anchor. Okay. The problem with this is that because these are small, you are very limited in space, so they don't have space for the giant weight, and you are very limited in energy, so they don't have the energy to carry around a giant weight. So they said, okay, we need you to make an anchor, but it has to be small, compact, and lightweight, right, which are all exactly the things that anchors are not. So, okay, so we thought, okay, well, let's think about that. And so we thought, all right, well, what if we had some kind of a technology that could actively burrow down into the sand and then set in some way, and then you send a signal, and it closes back up and burrows back out so that then you can move somewhere else. And it turns out that if you could develop that kind of a technology, there are a lot of other interesting uses for it. And one of my favorites is this one right here. So this is a picture from Life magazine. This is from the 60s. But in all honesty, the technology has not changed since then. So this is a boat that is laying cable. So this is how you lay cable across something like the Atlantic Ocean. So you basically there's a big coil of cable over here on the beach, and you tie it to the boat, and the boat goes out, and it just drags the cable behind it. Okay, the problem is that so then you drop the cable, it goes down to the ocean floor. But the problem is that you then have to bury it. You have to bury it under about 2 meters of sand. And there are many reasons why you have to bury it, but one of them is that if you don't bury it, the sharks are attracted to the electric signal and they come and chew on your cable. And so your cable doesn't last very long. So you have to bury this. And when you're in deep water, there are technologies to do this. And the technology is called a sled. So you basically have a sled that is tied behind this boat. The boat goes along, it drags the sled behind it, and that sled buries the cable as it goes. The challenge is when you're in shallow water. And shallow water, in this case, is typically 1 mile to 5 miles offshore. So if you're within 1 mile or to 5 miles offshore, this boat can't operate, right? So you have to do something else in this region, okay? And what you do is you get one of these guys. Like literally, you send a diver down with a shovel to bury 5 miles of cable underwater until you can get to the deep water, which is completely crazy. I mean, can you imagine that? It's hard enough burying cable on land. You have to put on a scuba suit and do this underwater? Yeah, no way. But imagine now that you could build one of these devices that I talked about where you have a little thing and it digs down and then it digs sideways and then it digs back up again. You could just attach 2 of them to this cable and have it follow along the cable and and pull it down under the sand. And this, by the way, is a roboticist's dream because your robots are attached to a cable, which means you can run power and control through the cable. You don't have to have onboard batteries. You don't have to have onboard control systems. Everything can go through the cable. So it's a fantastic problem for roboticists. Okay, so that's kind of the background of where we got interested in this problem. My student who worked on this is Amos Winter, so this is Amos. And Amos went through the biology literature, and he looked up about 200 organisms to see what kinds of strategies they used for digging and anchoring in sand. And the animal that he came up with is the razor clam. So this is a razor clam. This is a video I just took off of YouTube. So this is a razor clam burrowing into sand. You can see they're actually quite fast for a clam. So Amos likes to call these guys the Ferrari of bivalves because they are so fast relative to the other clams. Okay, so this is our little bivalve Ferrari. And the way it digs is as follows. So the clam has got a shell and a foot. So there's the shell and the foot. And this is the sequence of motions that it goes through to dig. So it starts the shell. So the shell sits like this. So imagine this is in the sand. The shell has a hinge on it, and when the clam relaxes its muscle, the shell opens. Okay, so if it relaxes its muscle, the shell opens. So you relax the muscle, so the shell opens and it braces itself against the sand. So while the shell is braced, it sends out the foot. So the foot comes out and it digs itself into the sand this way. Okay, then the shell closes. And when it closes, it squeezes the water that was in the shell into the foot, so the foot now blows up like a balloon, so you have a second anchor point, and then it pulls the shell down. So it's a ratcheting system where you have two anchors, and one anchor is the shell, which opened, and the other is the foot, which blows up like a balloon. And so you just ratchet your way down through the soil. Okay, and so this is also, if you're interested actually in the way animals move, this is a fabulous book here by Truman, which was written in 1967. So all of that was explained in Truman's book. Okay. So now, from a mathematical perspective, this is a fantastic problem. And it's also incredibly difficult because, so here's sort of the simplest thing you can imagine is imagine two spheres that are connected by some kind of a rod. And these spheres can expand and contract, and this rod can expand and contract. And again, you can make this sort of ratcheting system for this thing to move through the soil. Okay? But it's incredibly complicated because we don't know the constitutive equations for sand. We don't know how to switch well between fluidized and solid states, and sand can switch between fluid-like and solid-like behavior. We don't know what are the drag relationships for these things moving through sand. So there are a lot of things that are unknown. And so I'm not going to go into the details of that. Instead, I'm going to talk about the simplest scaling argument you could imagine. Okay, so what we really care about is how much energy does it cost you to dig? Okay, that's what I want to know is how much can I minimize the energy that it costs me if I want to dig? All right, so here are two things we know about granular systems. Okay, the first is that if you are going through a packed bed, so it's a packed bed of sand, the kind of sand you walk on, the force required to push something through that sand scales with the depth. Okay, and you know this because if you take a stick and you try to push it into sand, it gets harder and harder the further down you go. Okay, so that's what you're feeling, is you're feeling that force scaling linearly with depth. Okay, I know that energy is force times depth, so that tells me the energy scales like the depth squared. Okay, all right. On the other hand, if I think about a fluidized system, so let's say I have quicksand, or I have something sand and water, so the sand grains are not in contact, but there's a little bit of a lubricating layer between all of those sands. In that case, the thing behaves like a fluid, not like a packed bed. The force is now independent of depth, right? Because in quicksand, that's why you sink. The force is not changing. You just keep sinking. Okay, so that means that the energy scales linearly with depth. Okay, now the reason this is interesting is that I now have a signature that I can look for in clams and other digging systems. Right? So I can ask, if I look at how much energy they consumed, are they consuming energy at a rate that's going like the depth squared, or are they consuming energy at a rate that goes like linearly with depth? Okay, so that's what Amos did. Okay, and he did the following. So he wanted to look at both the packed and the fluidized case. So for the packed case, what he did is he took a clamshell and he glued it on a stick, and he called that his clamsicle. And he took his clamsicle and he just pushed it into a bucket of wet sand. And he just measure how much force do you require, how much energy is required to push your clamsicle into the bucket of wet sand. Okay, the next thing he did is he looked at the energetics of actual live digging clams to see how much energy they consume. Okay, so one of the things that I've learned in doing these projects is that biologists have measured everything. It's amazing what these guys have measured. So if there is something that you can put on a treadmill and put a little mask on it and measure the oxygen consumption, they have done it. So they know how much energy is consumed by any animal you can imagine. I mean, they've put on kangaroos and ostriches and cockroaches and whatever you can imagine. They've put these things on treadmills and measured consumption. It turns out they have not done this with clams because you can't put a clam on a treadmill. I mean, you can put a clam on a treadmill, but it doesn't do anything. Okay, so the question is, how do you measure the energy consumption of a clam when it's digging? So it turns out you can do this because we know all of these steps. And they have, even though they haven't measured the energy consumption directly, they have measured all of the pieces that you need for the steps. So they know the force that the muscle can exert. They know the pressure in the foot. They know how much the stiffness of the spring. They know all of these things. And when you put all those together, you can measure the energy consumption. So here's the answer that you get. Okay, so remember, what I want is I want a signature that either looks like energy scaling like depth squared or energy scaling linearly with depth. Okay, so this is the clamsicle. You push that into a bucket of sand, and you get something that looks like this, with a beautiful slope of 2. So the slope of 2 is the signature that this is going like depth squared. Okay, here's the real clam. So this is a live clam. The live clam, so you go through, you measure, you put in the energy from all these steps, and you get this line which has absolutely perfect slope of 1. So they are doing some trick to fluidize the sand around the shell to minimize the drag as they go through the substrate. Okay, so now this is great news. So the reason this is great news is that we have now uncovered the engineering principle you need to design a digger. Right? You don't need to make something that looks like a clam. You just need something that is going to open and close like this to get a pocket of fluidized material around the thing that's digging so it's easier to pull it through the soil. Okay? And so that's what we did. So we built Robo Clam. So this is Robo Clam right here, and this is the actual clam. Okay? Robo Clam, so we then took it to the beach to dig. And Robo Clam, when you actually put it together, this is the whole apparatus that you require. So this is Robo Clam right here, and this big thing is what you need to actually activate it. Okay? And the reason we need such a big chunk like this is that Amos decided that he was going to actuate this using compressed air rather than electricity, because he didn't want to be carrying around a giant power supply while he was wading around in salt water. So he thought, okay, so we'll use compressed air. So here's the compressed air, and now the question is, how do you want to actuate this thing, right? So here's at least the way you might characterize how you're actuating. So remember, there are a bunch of steps where this clam, so you open the clam shell, you push the foot down, you close it, you go down. So there's a bunch of steps where the shell is going out and in and up and down. Okay, so those are the different motions you need. You need out and in and up and down. Okay, and so here's the sequence of what you do. Since this is all compressed air, all you're setting is the pressure and how long you're turning that pressure on for. Okay, so for example, let's look at this. So this is the in step. So that's when the clam comes together like this. So all you need to know is, okay, how much pressure do I put in, and how long do I turn it on for? Okay, and what that means is that you just need 8 numbers. 8 numbers can completely characterize the kinematics of this thing, right? Because I need 2 numbers to describe this. I need 2 numbers to describe this, 2 numbers to describe this, and 2 numbers to describe this. Okay, so I need 8 numbers, which now finally brings us to our optimization question. Here's the optimization question. What is the optimal valving sequence that minimizes the energy required to dig? And this one I'm not going to ask you to solve, because it turns out this is actually very complicated. But so this is the question that we wanted to answer, and it's enormously difficult. And the reason it's enormously difficult is that we don't have any mathematical equations to describe what this thing is doing. Like I said, we don't know the constitutive equations for sand. We don't know how to calculate the yield stresses to find out where the division between solid and fluid is. It's a huge mess. I mean, the fluid equations are hard enough on their own, but when you turn this into a granular system, it becomes a disaster. Okay, so we don't have the equations, but we do have a robot. So here's what you do. You say, okay, I'm just going to choose some random kinematics. I'm going to let this thing dig, and I'm going to measure the energy consumption. Okay, and then I'm going to put an optimization loop around this. Okay, and what an optimization loop needs is it basically needs a black box. And normally what you put in this black box is equations, but we don't have equations. So instead we're going to put the robot in the black box. Okay, so what comes in is a guess at what we think are good kinematics. What comes out is a measure of the energy consumption, and so that we can use that to tweak our next guess, and so we can just keep going around and around until we find out what the optimal kinematics are. Okay, this is still an incredibly challenging problem because if any of you have done optimization problems, they tend to be very finicky. You can easily get trapped in local minima. They may not converge. It's a very, very hard problem. And now we've made it even harder because you now have to couple it to hardware. So now I have to couple it to hardware. I have to get the data in. I have to do this around. So Amos came to me and he said, okay, I think we should do this. And I said, all right, Amos, this is a really hard problem, right? I mean, I know Amos is a very ambitious guy, and I thought, okay, but I said, Amos, I think you need to scale this down again. This is not going to work. And he said, oh, no, I know exactly how I'm going to do this. And I said, okay, what are you going to do? He said, I'm going to hire an undergraduate to do it. What? Okay. So all right. Well, he's very enthusiastic. So I said, I'll say, but I don't want to hose an undergraduate. So I said, I'll tell you what, Amos, I will give you 2 weeks. I will find you a great undergraduate. You work with that student for 2 weeks, and then report back to me. Let me know how things are going. If it turns out this is just impossible, we're gonna shift and let the poor guy work on something else, because this is, I mean, really, you're not gonna make any progress on this. Okay, so we hired a student, Robin Deets, and Robin. So this is Robin up here. Robin was amazing. So this kid had this system, not only had he made progress in 2 weeks, he had the thing working in 2 weeks. Okay, and by working in 2 weeks, I mean he had to get the hardware working, he had to put the data acquisition card on there, he had to build all of the hardware to send this to the computer, he had to write the optimization code, he had to find the parameters for the optimization code. It is ridiculous what he did in 2 weeks, but this is the strategy that he took. So he said, okay, I'm going to use a genetic algorithm to find the optimal way to make this thing dig. Okay, and so the way it works is as follows. You say, okay, I'm going to start with a population of individuals that are randomly chosen. Okay, so here's my population of individuals that are randomly chosen. So each individual is some digging strategy, right? So here's a clam that does this, here's a clam that does this, here's a clam that does this. Okay, then I measure the energy consumption of each of them. Okay, and so that now there's some number that goes with each of these. Then I eliminate the weak individuals. So let's say those guys are bad. Then you combine the parameters of the successful individuals to spawn the next generation. Okay, so now I combine these 4 guys in some way that's, that whatever Robin worked out to make this thing work, and then you just keep repeating. And so you let this thing evolve, and you get successive generations. So the way this works is. And remember, this is how you tell if it's digging well. Okay, the signature we are looking for is an energy. If I plot energy as a function of depth, I want something with a slope of 1. Okay, this is what I'm looking for. I'm looking for a line with a slope of 1 on an energy versus depth plot. Okay, so what you do, you go in. So here is a random first guess. So Robin comes in, he puts in these parameters. This is his first guess. You plot the energy versus depth, and you don't get a slope of 1. You get garbage, right, as you would expect. This is a random bunch of parameters. Okay, so you get garbage. Okay, then what Robin does is he goes home. He has a nice dinner. He goes to bed. He comes back to the lab the next morning, and the next morning, the clam is doing this. It's amazing, right? I mean, look at this. This is almost exactly as efficient as the biological system. Okay, so using this genetic optimizer, you can actually get these robotic clams to dig as efficiently as the actual biological clams. So we're now working on the next step to try to commercialize these with bluefin, and this is just a picture of Robo Clam digging in his native environment off of Cape Cod. Okay, so that's the clam story. And then the last story I'm going to tell you is about tiny swimmers. And I'll warn you ahead of time, there's no robot at the end of this one, but I think it is kind of a fun answer that we get. So tiny swimmers. So the first thing I have to do to talk to you about tiny swimmers is we first have to define what I mean by tiny. Okay, so in fluid mechanics, the thing that describes the scale of whatever you're looking at is the Reynolds number. Okay, so here's the Reynolds number. So the Reynolds number tells you the ratio of inertial to viscous forces. Okay, and so this is the definition of the Reynolds number. Hopefully some of you have had fluid mechanics classes. And this is the density of the fluid it's swimming through. This is the viscosity. This is the velocity of the swimmer. And this is a characteristic length scale of the swimmer. Now, In biological systems, this actually becomes much simpler because biological organisms tend to swim in water or in fluids that have properties that are very close to water. Okay? So what that means is that I cannot change the density or the viscosity. So this Reynolds number is set entirely by the size and the speed of the organism. Okay? There's another kind of rule of thumb in biological systems that says if I want to estimate how fast a thing can go, and this is very, very rough. In biology, there's exceptions to everything in biology, so this is very rough. But you could estimate roughly that how fast a thing goes is basically its size per second. Right? So I'm about a meter, so I can move at about a meter per second. Right? Ants are about a millimeter, so they move at about a millimeter per second. Paramecium are about 100 microns, so they move at about 100 microns per second. It's very rough, but at least gives you an idea. So you can use that to estimate the Reynolds number for various systems. And so just to calibrate yourself, I'm going to now show you the Reynolds number for a couple of different things, starting with a person. Okay, so a person swimming in water, you can think for a minute whether you think this is going to be high or low Reynolds number, whether you're dominated by inertia or viscosity. So a person swimming in water has a Reynolds number of about 10 to the 5th. Okay? Now, if I put up a whole scale here of other things, so tuna fish over here, these are at about 10 to the 6th. As I go down in scale, ducks are maybe about 10 to the 3. Ants are above a Reynolds number of about 1. Okay, now my plot turns this corner, and it goes up this way, so this is going from big to small over here. So here's a paramecium. Again, this is kind of comparable to the scale of the ant, so it has a Reynolds number of about 1.5. So, and I'm interested in low Reynolds number swimming. So low Reynolds number swimming means that I'm interested in this kind of region over here. So this is the region between bacteria. So this is a bacterium here, which is about 1 micron. And sort of single-celled organisms, which are between 10 and 100 microns. Okay, so this is just to give you an idea of the kinds of swimmers that I'm talking about. So I'm interested in what's going on over here. And again, just to calibrate yourself, this is approximately the smallest thing you can see with a light microscope, right? Because this is at 1 micron, and the wavelength of light, green light is, 600 or 700 nanometers, something like that. So, you can play tricks to go smaller than this, but roughly for a light microscope, this is about what you can see. Okay, now the interesting thing about looking at low Reynolds number, so one of the important things about this plot is notice that all of our intuition about the way fluids move is set over here at high Reynolds numbers. Okay, and what that means is that we can be very much led astray by our experience in what we expect fluids to do. And there's a very interesting example to show how wrong we can be, which was done by G.I. Taylor, who was one of the giants in the field of fluid mechanics. Okay, and G.I. Taylor did the following experiment. So he put 2 concentric cylinders together, and he said, okay, I'm now going to fill this gap between the 2 cylinders with a very viscous fluid, okay, and I'll put a blob of dye in that gap, and then I'm going to mix that dye in by spinning the inner cylinder. Okay, so very simple experiment. Okay, we now have a movie of Taylor's actual experiment, of G.I. Taylor doing this experiment. It's in the National Committee for Fluid Mechanics Films, because apparently we had a National Committee for Fluid Mechanics Films at some point. Okay, and so I'll just show you the movie. So this is G.I. Taylor's hand that you see. So this is the concentric cylinders. This is the edge of the other cylinder. So the gap is only about this big, right? This is the rod in the middle that turns it, but the gap is sitting something like this. So he injects a blob of dye. So I think the fluid he has in here is glycerol, which is about 1,000 times the viscosity of water. Okay, so that's how you know it's low Reynolds number. And now he's just going to mix in the dye. He's going to do it fairly slowly because I want to keep this low Reynolds number. So you want the velocity to be slow. So so he's mixing the thing in. He turns it, about 4 times, something like that. And now for good measures, he's going to mix in the other way. So so now he's spinning the inner cylinder. And we have to be a little bit patient here. So spin it around. I don't know what that was, 2 or 3 times. And he comes back around. And the thing is unmixed. That's totally real. Okay? So this is how your intuition can fail you when you think about low Reynolds number flows. They do things that are very different than high Reynolds number flows. Okay, so and this effect here, this is called kinematic reversibility. Kinematic reversibility is a disaster for low Reynolds number swimmers. So if you're tiny, if you're a bacteria, you are subject to this, that things are reversible. So if you can do this, that means that you do this and you move over here, okay, and then the only thing you can do is this, and you go back exactly to where you started, right? So you are not going to be a very good swimmer because all you can do is go back and forth. So there are lots and lots of strategies for swimming at low Reynolds number, and this is a field that's well studied now. There are many, many people that are thinking about what is the right way to swim if you're a low Reynolds number swimmer. But I want to focus on biology. So I want to tell you about what biological systems actually do. Okay, so I'm now going to put up 2 slides on biology. I am not a biologist, so the extent of my knowledge of biology is going to be in these 2 slides. Okay? And my first slide that I'm going to put up is a fluid dynamicist's view of biology. Okay? So this is what biology looks like to a fluid dynamicist. It's confusing and messy. Okay, so this is a slide. So this is a slide that was taken from a lecture by James Lighthill. And James Lighthill was sort of one of the pioneers in the study of swimming organisms, fish, microorganisms. And so this is obviously the days before PowerPoint when you had to draw your slides by hand. So this was literally the slide that he drew in his lecture on flagellar hydrodynamics. And the reason I like this portrayal is that Lighthill has organized his swimmers, not by any kind of biological metric. He's instead organized them first and foremost by how they swim. Okay, so here, if you look in this circle, roughly everything in this circle has got a head and a tail, or a head and 2 tails. Right? That's pretty much what's in this circle. So they all kind of have this geometry with either 1 or 2 tails. Okay. The next sort of level of breakdown here is he's divided this between prokaryotic cells and eukaryotic cells. Okay, so if you think back to your high school biology, prokaryotic cells are basically bacteria, and eukaryotic cells are everything else. So us, algae, penguins, whatever, they're all eukaryotic cells. Okay, so this line over here is the dividing line between the prokaryotic and the eukaryotic cells. And this turns out to be very interesting because the mechanism you use to swim is very different on either side of this line. Both have been well studied, but the one I'm going to talk about today are the guys down here, which are the eukaryotic cells. Okay, so the eukaryotic cells, again, I'm interested in basically things that have a head and a tail, and for the purposes of this talk, I'm only going to think about 1 tail. Okay, so that's my first slide on biology. My second slide on biology, I now have to tell you something about this tail, which is the primary mechanism that these things use to swim. Okay, so here's how the tail works. If you take a cross-section of the tail, you find something that is called the 9+2 microtubule structure. Okay, and the 9+2 microtubule structure means that the cross-section of all tails looks like this. It has 9 pairs of these little tubules. So imagine these the tail is coming out this way, right? So these are long tubes, okay? And so there are 9 pairs with 2 in the middle. Okay? And this is something that I find incredibly remarkable because it is the same in all species. Right? There's nothing in biology that is the same in all species. Biology is a mess. There's always an exception in biology. Not true with this. There is no 10+3 microtubule structure. There's no 7+4, none of that. It is always 9+2. And I don't think anybody actually knows why it's always 9+2. But if you look at the cilia in our lungs, it's 9+2. If you look at the tails on swimming green algae, it's 9+2. If you look at zebra sperm cells, it's 9+2. Anything that is a eukaryotic cell with a tail has got this 9+2 microtubule structure. Okay, and what that means is that the diameter of all of the tails is approximately constant across all species. Everything. Us, algae, kangaroos, grasshoppers, whatever. It's all always the same. Okay, so that's the first thing you have to know. The second thing is that because it has this structure, these organisms can apply local bending moments along the tail, which means that they can pick the shape. So they basically take these pairs of microtubule structures and slide them relative to one another. And if you slide them relative to one another, then that gives you a local bending moment, so you can make this thing do waves or whatever you want, whatever shape you want to do. Okay, so that's all you need to know about biology for the rest of the things I'm going to tell you. All right, so small swimmers. So this was a project done by my student Daniel Tam, and so Daniel decided, for starters, that he was going to think first about sperm cells. And the reason he decided to think about sperm cells is because the objective function on sperm cells is incredibly well defined, right? The sperm cell only has one goal, and that is to move genetic material. Right? They don't have to find food. They don't have to communicate with other sperm. They don't have to do. They don't have to none of this. All they have to do, they have a packet of genetic material that's in the head, and their mission is to move it. That's it. Okay, so it's purely a transport phenomenon. And the question Daniel asks is the following. For a given head size, so the given head, so this is where all that genetic information is packed. So I have some head which is holding all that genetic information. What is the optimal tail length? Okay, again, that's our optimization question. Okay, so he made a beautiful model, and you can calculate things like the swimming efficiency for a given tail length and a given head size. So what this plot is showing, these are lines for different head sizes. So this is one head size, this is another head size, this is another head size. And what Daniel did is he said, okay, let's pick a head size, now let's pick a tail length. So let's consider that point right there. So that's got some head size, whatever it is, and a tail length of, I don't know, 120 microns or something like that. Okay, for that particular geometry, that's tail and head size, he then ran a functional optimization problem to figure out what is the optimal way to move that tail around, right? So should it be flapping around like this, or should it be flapping like this, or should it be flapping like this? So he ran the functional optimization problem to figure out how this thing should swim. And he ran that on each one of these points. So there's a huge amount of calculation that goes into this point because each one of these points is a functional optimization problem. Okay, so once you do that, you can get this very nice plot. And then you look at it and you say, oh, okay, well, actually, you know, we should have thought of this, that there's really only two length scales in the problem, the diameter of the head and the length of the tail. So if I normalize the length of the tail by the diameter of the head, all of these things should collapse onto one curve. Okay, so he did that, and they do. So it's beautiful. Okay, so you get this. So they all collapse onto one curve. So this is the efficiency versus a normalized length of the tail. So Daniel brought this plot to me, and this is possibly my favorite plot of all the plots that my students have ever brought to me. Okay, and the reason I love this plot so much is that it has an answer. And the answer is 12. Right? The answer right here. Here's the answer. So I said, Mike, what was the question? The question is, what is the best possible swimmer? So I say, okay, I need to optimize efficiency. Here's my efficiency. There's the optimal efficiency. Okay, where does that happen? Oh, it happens right down here at about 12. Right? So the answer is that regardless of species, regardless of material, regardless of what this thing is doing, the optimal ratio of tail length to head size is going to be 12. Okay, so the next thing Daniel did is he said, okay, he went into the biological literature and he looked up the ratio of head to tail sizes for 400 different species of mammalian sperm. Okay, and he made a histogram of these guys. So here's the histogram of the 400 species that he looked up. And the answer is 12. It's 12. It really is. Right, so this was amazing. So I was super excited about this plot because we've really rationalized something from very fundamental hydrodynamic principles about what you observe in biological systems. Okay, so I went around. I was giving this talk everywhere. This was a great talk. And one of the things that I learned when I was giving this talk is that physicists and biologists see something very different when they look at this plot. Okay, so here's when a physicist looks at this plot, they say, oh, look at this. The answer is 12. There's a whole pile. There's a beautiful peak at 12. We have now understood something fundamental about this system. We have rationalized something about biological systems. That's great. Biologists, when they look at this plot, say, this peak is boring. Everything is in this peak. The interesting thing in this plot is the outliers, because the outliers must have been subject to some other kind of evolutionary constraint or optimization. Otherwise, they wouldn't be outliers, right? So first of all, I thought that was very interesting to think about the different the different philosophy of physics and biology, which totally makes sense, right? Because in physics, you become famous when you unify everything. You know, as you saw in Tony's talk the other day, everybody's shooting towards those really initial moments when everything is unified. That's how you become famous as a physicist. As a biologist, you become famous when you discovered something that nobody's ever seen before, right? Which is totally different from everything, right? And that's what they see in this plot. All right, so having done that, I was brought up as a physicist, so I was excited about this peak, but I realized, okay, well, actually the biologists have a good point. These outliers are probably doing something interesting. So we decided to look at the outliers, and so in particular, let's take this one, which is the furthest outlier, okay? I want everybody to think about what you think that outlier might be, okay? Remember, these are all mammals, right? These are all mammalian species, so I want you to think now for a moment, what is the weirdest mammal you can think of? Okay? All right, so I'm now going to tell you the answer. Everybody has a weird mammal in their mind. Okay, so here's the answer. That point is the bandicoot. Okay. Now, the bandicoot is a marsupial. So anybody who guessed platypus gets full credit. Okay, so, but the bandicoot is a marsupial. And so then you ask, okay, well, why is the bandicoot an outlier on this plot? And so we do a little digging, and we do a little research on the bandicoot, and we find the following. So remember that I showed you a few slides back. I said, okay, this is the microtubule structure. Everything is a 9 + 2 microtubule structure, so everything, the diameter of the tail is constant across all species. Okay, what I should have told you is that the diameter of the tail is constant across all species except for the bandicoot. Okay, and here's how we know that the bandicoot is different. So the bandicoot is different because there was a paper, we know it's different because there was a paper that was written in 1958 by Cleland and Lord Rothschild on the bandicoot spermatozoan, an electron microscope study of the tail. Like I said, biologists have measured everything. It's incredible. You look for it, they have every piece of information you could possibly want. All right, so here's what they found. Here's the electron micrograph that they got. And the bandicoot, it still has the 9 + 2 microtubule structure. That's true. Still, everything still has a 9 + 2 microtubule structure. But in this case, it's encased in something called a sheath. And this sheath actually makes the diameter about an order of magnitude larger than it is in all other tails. Okay? And when you make the diameter an order of magnitude larger, that means you make the bending moment, the bending moment of inertia larger by 3 orders of magnitude because it scales like r cubed. Right? So suddenly there's an enormous cost to bend this thing that we didn't have before. Okay? And so when you put that cost in, it turns out that shifts this peak in this direction. And so again, this turns out to be optimal when you put in the correct radius of the tail. Okay, so we understand that outlier. I have one more thing to tell you, which is that there's another group of outliers on here that I don't understand why they're off. But so I'm going to leave you with a puzzle. Okay, and the puzzle is these guys right here. Okay, so these yellow points over here, I should tell you, so the colors in this histogram represent different orders, right? So there's rodents, there's bats, there's carnivores, okay, so colors are different orders. Okay, and so you can see that, most of them are kind of centered around this 12, right? There's some scatter in different directions. But over here, there's an order that is systematically off. And there are a lot of points in here. There's about 100 different species in here. Okay, and they're off by a lot, right? Because they're sort of closer to 6, right? Which is nowhere close to this optimal peak. So I don't know why they are off, but I know what they are. So let me show you what they are, then you can think about why you think they might be off. So those guys are the even-toed ungulates. So they are hooved animals. And like I said, I don't know why they're off. I mean, one of the things that this analysis has done is it has singled these out, so it makes them now an interesting point to study. So that's what I have to tell you today. I just wanted to put up one more slide to thank everybody who's involved. So, of course, the Clams, Amos Winter was the lead graduate student on that, and, of course, Robin, who did the fabulous optimization, and Sonny Young, who's now at Virginia Tech, also helped us out with that. Snails, Brian Chan, Neil Baumforth at UBC, Eric Lago, who's now at Cambridge, and John Wilkening at Berkeley, and the tiny swimmers were done by Daniel Tam. These are all our funders, and if you want to learn more, this is my website, hosoigroup@wordpress.com, and I thank you all for your attention.
I think we have time for a few questions.
I stumped them.
No, you did. Well, just to begin, I'd like to thank you for making crawlers and diggers and swimmers speak to us in such eloquent terms, and for showing the interplay of all the different sciences. It was just a beautiful talk. It's sort of a 2-hour talk given in 1 hour, but it was perfectly clear.
Thank you. I think we have a question.
Here we have.
Yeah. Hi.
Thank you for the wonderful talk. My question is, 500 years from now, do you think aeroplanes will fly like birds?
Oh, good question. Okay, so I'll repeat the question. So the question was, we're going to work on the microphone. The question was, 500 years from now, do I think airplanes will fly like birds? And I think that's a really good question because it depends on what you mean by aeroplane, right? So if you mean the planes that we see today that are carrying around hundreds of people, I think it would be very hard to make them fly like birds because of the stresses in the wings. Having said that, there are lots of applications nowadays for small-scale airplanes, like drones, that kind of thing. that where you're looking actually at the scale of birds, I think you could easily imagine a flapping device that could perform as well as the biological systems.
Thank you. Great question.
Yeah.
Any others? We have a question over there.
Oh.
I'm just wondering, what are the next biological systems that you're thinking about looking at, and how do you decide what's next?
Oh, yeah. Another great question. So the thing that I'm actually getting really excited about right now is plants. And the reason I'm excited about plants is, again, because I like things that are as simple as possible, so I don't want to have to think about a nervous system. And so what could be better than plants? There's no brain, right? And there's no muscles, so it makes it much easier. So we're actually thinking about pumping and digging from the point of view of plants. So plants are actually very good diggers because their roots can penetrate through all different kinds of soils, through rocks. So we're looking at digging and then also pumping, because a tree that is 100 meters tall actually has to pump a huge pressure head. And they have to do it with no moving parts. So how do you pump enormous pressures with no moving parts? So we got very interested in plants. As far as how we choose, it totally depends. So a lot of the times I let my students guide me in which direction we want to go. Sometimes we choose based on what is the application. So we have companies that come to us, they say, we need to do something like this, and we start looking at biological solutions and let that guide us. Sometimes, though, students are just interested in things. So I have one student right now who's interested in the thermal properties of Arctic mammals. Right? And so that's actually a really interesting problem to think about. Can we design clothing or wetsuits that have the sorts of efficiencies that you see in polar bears and penguins? So it varies.
Thank you very much for the beautiful presentation. I've always been intrigued by how nature inspires us in modern technology, or how we realize that our modern technology looks very similar to nature. But I always wondered, can we, with models that simulate evolution, can we outpace nature? Mm-hmm. And you seem to have answered that by optimization problems.
Yeah.
So do you think that, for example, clams in the future will look more like your robots?
Yeah. Oh, yeah, great question. So it turns out actually the Robo Clam looks pretty similar to real clams. So we kind of converged the biological solution. But I love that question, because one of the things that we could do to actually do better than nature is use the fact that we don't necessarily have the same constraints. Right? Because clams, digging is not the only thing they have to do. They also have to forage, they have to eat, they have to meet other clams. Right? Whereas if I'm working in the oil industry or if I'm working in the construction industry, all I have to do is dig. Right? So I don't have all of the same constraints that the biological systems have. And because of that, I think you can actually do better. The other thing that allows you to do better is we are also not constrained by biological materials. Right? So there are materials that can withstand stresses that are better than what you see in biology. And so if you're allowed to use those, I think you can also outperform some of the biological systems. Great question.
We have a question over here from one of our artists in residence.
Oh, excellent. I love artist questions.
Well, being an artist, I'm interested in frequencies. With the clam robot, was it considered using frequencies to liquefy the sand?
Oh, great! Absolutely great question. Yeah, so the frequency makes a huge difference. And one of the reasons clams can do what they do is that they are digging in fully saturated soils. Okay, so that means that the interstitial fluid is water and not air. And so when you fluidize, it takes longer for the particles to settle because the water is retarding that settling. So if you took that same clam and you had to dig in the desert, so it now had air as the interstitial fluid, the particles settle, that you lose the fluidization much faster, which means that the frequency that you have to operate at, you have to operate at a much higher frequency in dry sand than you do in wet sand. So the frequency is sort of determined by the environmental conditions.
I guess what I meant was sound frequencies.
Oh, sound frequencies.
Yeah.
Oh. Sorry, say it again. So, as in...
Using sound to liquefy.
Oh,
sound.
yeah, using sound to liquefy. Oh, which you could totally do. So I don't know of clams that do that. I'm trying to think if I know of any other organisms that do that.
Well, I guess in the topic of inventing something that is beyond nature.
Yeah, that's a great idea. That's a really great idea. Yeah, we haven't tried it.
Quick, lock the doors. No one
Yeah.
leaves until we patent
Yeah,
that.
exactly.
Okay. Well, you've made many things beautiful to us, and to me you made mathematical modeling as beautiful as ever tonight. And we have a beautiful artifact of the ancient Arabic world for you, which should not be a surprise because you've been here for a few
For the
of
other
these
two talks.
now. But we have an Arabian astrolabe,
Oh,
a
thank
replica for you, and a web
Wonderful.
T-shirt and
Oh,
a
wonderful.
clicker for, we hope, hundreds more of these talks that put very complicated phenomena within reach.
Yep.
And another thing that's beautiful about your talk is the way you and your students communicate
Yes.
and pick problems, and I think that's been an inspiration to all of us who are either faculty or students.
Great.
So let's thank Pecco tonight.
Thank you.